Hey everybody, Mr. Bruss here, your favorite Exploration Academy science teacher. And I just wanted to give you a quick overview of the DNA seminar. I know for those of you that were in class yesterday, we went over this a little bit, but I just kind of wanted to recap that. And for those of you that weren't able to make it to class yesterday, to kind of just get a sense of what the purpose of this seminar is. So I'm going to go over into Canvas. So the first thing is in order to do this seminar, you should be able to get into Canvas. And so I made a page, a module, however you want to call it in Canvas for EA. And if you're not in that yet, let me know and I can get you there. So I'm going to start sharing my screen from the iPad so you get a little bit more perspective of what it looks like from your view. So the first thing you want to do is go into Canvas here. So we'll load up Canvas. And right here is this EA Science. I put together this Canvas course for all of the different seminars that we're going to try to do this year in EA. So click on that. And the first thing you'll see is I'm still kind of building out or editing the exact dates. But if you look at this one, this is Seminar 1, Unlocking the DNA Code. And I've gone through and I put all the days. So there are 14 total days plus the day of the assessment. So 15 days. And I'm starting to fill this back in with dates, but I think you can probably figure out the order that this is going. So what's the point of this seminar? What are we trying to do? And the first thing is if the first thing that you need to do is we're going to kind of complete a project along the way that's going to teach us how to do the assessment at the end. The assessment at the end is identical to the project that you're going to be working throughout the seminar, just with a different nucleotide sequence. So what does that look like? So first, if I click on this student formative assignment list. So everyone in the seminar is going to get their own specific nucleotide that they need to unlock as a part of practice. And then you'll have a totally new one. Oh, what the heck? Oh, there I am. You'll have a totally new one that you'll get to try to unlock for the assessment. So here I am right here. Brussy Be Cool. And I've been assigned nucleotide HS17. So if I click on that, it's going to let me click on there. Oh, open link. Sure, let's open that up. Let's see what happens. So if I open up this link, sure, we get all kinds of folders popping up. And here I am. I don't want to do anything with that. Can I untap that? There we go. DNA nucleotide structure HS17. And what I have here is a sequence of nucleotides. There's actually 21. You'll see that I add that, and that relates to having exactly 7 codons, which you'll learn about throughout the seminar. So I only have 20 here, but there are 21. And as I move down these slides, you can see that the first thing I need is each of these nucleotides that I need to encode is highlighted right here in blue. And it tells me that this is the first nucleotide in a 5' to 3' DNA nucleotide sequence. So I need to determine what structure that is, which of the 4 DNA nucleotides that is. It happens to be A, adenine. I can tell because it has a double ring structure and only has 2 hydrogen bonds to thymine here. The next one is position 2, looks like A again. Position 3, I can tell that that's C right off the bat, and so on. So your very first task in this seminar is learning the skills of being able to identify the nucleotides in your mystery sequence. Okay, so once we kind of have that, let's hop back over into our modules here. And I can now look at the assignment as a whole. So here's our formative project. Formative means like teaching, right? Summative is like kind of the test at the end. So here is the project that we're going to be working on. And The overall, so the kind of the essential question is, how do 4 super simple molecules combine to make all the different structures of life, to give the code of all the different structures of life? And we're going to use a string of molecular structures, those are the nucleotides we just looked at, to determine the sequence of nucleotides. Then we're going to figure out the gene that it contains, this unique string of nucleotides, the amino acid sequence, and maybe at the end we'll describe the protein function. So here I've given you an example slideshow, but it's just like the one I showed you. And there are 5 tasks in sequence here. So our first task is to identify the nucleotide sequence. That's just what I was going through right there. And then to write the complementary sequence. So remember, DNA is double-stranded. So there's one sequence and another one kind of aligned with it, which is called a complementary sequence. And that is right here. So here is task 1, is to be able to look at that black and white chemical structures that I showed you and be able to figure out that nucleotide sequence, and then from there, figure out the complementary strand. So we're going to learn those skills in the first couple days of the seminar. The next task is to now transcribe DNA into RNA. And you haven't learned this yet in the seminar, but RNA kind of carries the message of DNA out of the nucleus so that it can deliver the message of what protein should be built, what amino acid sequence should be put together. So that is task 2. From what you'll learn is from this DNA sequence, you can transcribe and make it into mRNA. So we'll learn that as well. The next thing that we're going to do then is from this mRNA sequence, mRNA is read in 3 nucleotide words, I guess you can call them. They're called codons. And that tells us what amino acid should be put together. So if there are 21 RNA nucleotides, that's going to code for 7 amino acids. So there should be 1, 2, 3, 4, 5, 6, 7 positions here. So that's part 3 of the seminar. Now, part 4 is, I'm going to give you somewhere in your DNA code, I'm going to say there's a mutation. A letter, maybe it was an A got switched to a T, or maybe a whole new letter got added, a C or a G was added. Now you have to retranscribe what would the new mRNA look like based on that mutation. And then you have to say, would this code for new amino acids? Many mutations change the amino acid code, but not all. So you're going to tell me, you're going to code the new amino acids that happen after the mutation, and you're going to tell me how it's changed down here versus what you coded in part 3. So now we're 4/5 of the way through, and the final thing we're going to learn is how to use a program called, a tool called NCBI BLAST. So this is the National Center for Biological Informatics, Biological Information. And BLAST is this tool where you can take any string of nucleotides and search every single DNA sequence that has been uploaded to this database of all different kinds of species. There's like T-Rex species in there. There's blue whales. There's bacteria, there's viruses, and for our purposes, there are humans. And so you're going to put in the sequence that you've uncovered from part 1 into this database searching tool, and you're going to tell me what gene that sequence is a part of. And I've made, actually for everyone, I've made a real human gene that does something in the body. It might be an enzyme related to DNA replication. I made ones that make fatty acids, ones that make cholesterol. These are all real genes. And I try to pick out ones that either had funny names or were really important for health and disease. And then from there, you can also tell me what chromosome that gene is related to. All of that comes from that NCBI BLAST tool. So That is the overview of the seminar. So along the way, what we're going to do, I'll just recap the overview, is each day or two, I'm going to teach you how to do, I will, and the videos that are provided for you are teaching you how to do these 5 tasks right here. And then you get a chance in this formative assessment, remember with your own unique nucleotide structure, you get a chance to practice in this formative assessment each one of those tasks so that you feel prepared for the final assessment where you're going to get a brand new sequence and you go through these 5 steps. And my hope is that when you know how to do this, you can go through this relatively quickly. You'll be able to read the nucleotide code pretty quickly, understand the complementary strand, be able to transcribe RNA, read a codon table or codon wheel to figure out the amino acids, do that again with mutations. And then here, this is just a simple internet search using that tool, and you'll be able to read the results back from that. So I'm hoping that this is pretty straightforward, and it is like what scientists do, where we're trying to solve problems and not necessarily just memorize structures, but to solve a problem. And in this seminar, in this unit, your problem is to really identify the gene that a certain string of nucleotides belongs to. That's all of it. So I'm hoping that this will be interesting to you all, kind of a new way of looking at what you can do with DNA and a lot of the actual scientific tools that are available. So I look forward to seeing you all in class, and I'm hoping that this goes well for everyone. All right, bye-bye.